Hi everyone, my name is Mark, and today we're going to be talking about statistics and research. I also have my other presenters here with me.
Hey, hello everyone. My name is Brett. I'm an ambassador for the Center for Undergraduate Research, and I'm also one of the co-hosts for today's workshop.
Hi everybody, my name is Kavita, and I'm also one of the co-hosts for this workshop and a co-ambassador as well.
So the layout of this workshop is basically going to be that we're going to talk about the experimental design portion of statistics. We are then going to go into some inferential and descriptive statistics. And following that, we're just going to do a brief overview of two commonly used statistical softwares, as well as a short demonstration of each of them. So to start with, we're going to talk about observational studies. An observational study is when a random sample is obtained from two or more populations, and then the outcome is observed and compared across those two groups. One of the downsides of an observational study is that it lacks the ability to define a causal relationship, and confounding variables can also influence the data. A lot of times what you hear also is that correlation does not equal causation, and this can be applied to an observational study. Additionally, we have case studies, which is an in-depth look at a particular person, group, subject, or situation. This is often, if anyone wants to go ahead and mute themselves too, just make sure throughout the presentation, just can everyone see the slides right now?
If anybody can't, please let me know and I will fix that if you want me to here. And this is better.
Okay, I think we're okay.
If you can't, just send me a chat.
Okay, I think we're okay. So once again, going back to a case study, it's often used to provide context or background on a very specific issue. And a lot of times people see this in the field of medicine whenever there's an individual patient with abnormal circumstances or an extreme medical case that doctors want to highlight for other physicians to see. On this slide, we just have a few of the experimental vocabulary that you'll typically see in statistics. So first off, there's a factor, which is just the variable that's being studied within your experiment. Additionally, there's a treatment, which is just a particular subform of the factor. And the goal of the treatment is that the only difference between the subjects within the experiment is the treatment they are given. And this is how you can decipher between if the subject within the experiment is getting the treatment or has no treatment applied to them. And this allows for a cause and effect relationship to be determined within the actual experiment. Next, we're talking about basically using a completely randomized experiment. This is almost always used in all types of experimental data. It's seen as the gold standard, and this is because every single subject within the experiment group is randomly assigned to their different treatment levels or groups, and it allows for an equal number of the subjects in each of the group, which allows for less bias and also increases the randomization of the statistics that are collected from the actual experiment. And then another type of experimental design is a randomized block design. And this is used when there's a variable that can confound with any of the data that they're trying to collect, and the experimenter typically cannot control that. And this is commonly seen in age, sex, or experience. And within each of the blocks within the randomized block design, the subjects are randomly assigned to the treatment or the factor treatment levels. And basically, the experiment is conducted separately within each of these blocks. And one other thing that isn't here, one of the things that you'll see in a randomized block design is something known as age or sex matching. And this is in order to make sure that you have an equal number of people who are getting the treatment and people who are not getting the treatment within each block who have those specific characteristics, like having the same age or both being male, both being female, or whatever sex they identify as.
So I am going to briefly discuss considerations in experimental statistics. So one of the biggest things that you see in experimental statistics is blindedness. So that is when the experimental subject or the experimenter themselves do not know the treatments the subjects have been assigned to. So there are two types of blindedness that occur. Either they opt to do a single blind, which is where only the subjects do not know which group they are being put in, or even more effective would be a double blind where the subjects and the researchers do not know which group the subjects are in. And this is extremely important in reducing any experimenter bias within your experiment.
So one thing I didn't mention at first was if anyone has any questions, you guys can just go ahead and say anything in the Zoom chat, or if you just want to raise your hand, or even if you just want to interrupt a little bit, you can ask a question, or if you just want us to elaborate on something. And then also at the end, we'll be here for questions if anyone wants to talk about research or just statistics in general. So here on this slide, we're talking about some descriptive statistics, and the first one that we're talking about is accuracy, and this is how close a value is to the expected or theoretical value. To the right there, we have an archery board, and I believe it's a really, really good representation of showing the difference between accuracy and precision. So the center of the board is what you want as your target or your theoretical value. And whenever you have a high accuracy, that means that you are getting as many as you possibly can in that very center hole. And then if we move on to the idea of precision, this is the similarity of repeated measurements, or basically the repeatability of what occurred during the experiment or the trial. In this case, these people are shooting arrows. And as you can see in the top left of the graphic, there is low accuracy and low precision. Because the fact that there is only one accurate arrow in the center, and then none of them are clustered close together, this shows that there's only one accurate shot, and they're not very precise because they're extremely spread out. Whereas if you look in the top right, there's a very, very close cluster of all of those arrow shots, but there's a low accuracy, and it just helps to show you the fact that you can have high precision and you can have low accuracy at the same time, or vice versa. So next we're going to talk about some of the measures of location. These are mean, median, and quantiles. I am sure you guys have heard of the mean or the average, and the median is just the very center value of your data. And then a quantile is just, it contains X% of data to its left. And these are all just very commonly used terms. And then there's also measures of spread, such as standard deviation or variance. And finally, there's something known as the 5-number summary, which includes your minimum, your 25% quantile, the median, the 75% quantile, and the maximum. This is commonly seen in a box plot, and we will actually see that on the next slide as it shows you exactly what is going on with that 5-number summary. If you look at the top right there, those are called box plots or box and whisker plots. The whiskers are seen as those range of values which contain the max and the min. And also you can see that there are little circles that are above and below the data there. Those are highlighted because they are outliers, and typically, very frequently in statistical analyses, you want to remove your outliers, but you still need to acknowledge the fact that they are there. And then that middle line in the middle of the box plot is actually showing your median value. And then the two less bold lines surrounding it that actually form the block are the 25th and the 75th quantile. And the data in between those two bars is actually the middle 50% of your data. And then below that chart, we have a histogram, which helps to show the visualization of frequency. The x-axis represents the ranges of possible values that you can obtain during your experiment. And these data are split into different bins based on their frequency. And then the y-axis helps to determine how often or how frequent something within that bin is occurring. And to the right there, it's just showing a simple frequency chart of how often a number or the data that you're collecting was within those X value ranges.
Okay, so I'll be talking about inferential statistics, which is essentially the follow-up analysis after you get your descriptive measures of your data. And inferential statistics essentially comes in two forms, which are confidence intervals and hypotheses tests. And I'll be talking about confidence intervals right now. But essentially, whenever you report a figure to a scientific journal, it's not enough just to provide your estimate from the sample due to the inherent randomness in your sample. Because if you continue to take samples over and over again, your estimate may not be the same each time. So you should provide some type of estimate of the uncertainty in that value. And in order to do that, we generate a confidence interval to try to capture some of that uncertainty. So for instance, if you're doing a drug study to find the effectiveness of a certain drug, you may find in your sample that it's 15% more effective. But in a different sample, it might be 20%. So you want to find a range in which that true value that you're looking to estimate really is in. So a confidence interval is just that. It's that interval in which the unknown value that you're looking to estimate with your experiment falls in. And you set the confidence based on, as the experimenter, the type of tolerance you're looking to get. As an extreme, you can make a 100% interval, which is just the entire real line, which will 100% contain what you're looking for, but it provides you no information. So you can shrink the intervals and make them smaller, where you risk missing the data you're looking to capture, but it would be more precise as it's smaller, and it gives you much closer of an idea of the spread of the data. The way the language we use is that there's a C% of the new intervals produced will contain that unknown value we're looking to capture. And by convention, that is used as 95%. So 95 out of 100 intervals should theoretically contain that unknown value we're looking to capture. So confidence intervals are made up of two components primarily. You have your point estimate, which is just the value you obtain from your sample, plus or minus your margin of error, which provides the width of the interval. So on this slide here is a visualization of what confidence intervals mean. If you look to the right, there's an orange dashed line going through the value of 0. So in this case, the unknown truth of the mean for the entire population is 0. But of course, we can't actually measure the entire population. We can only measure a few samples at a time, just out of convenience sake. So we don't really know what the true mean is, but we're trying to approximate that using our sample. So in each of these black lines, those are the confidence intervals for a new sample taken, and the blue dot is the sample mean. And for the intervals that across this orange line, that means the value of 0, which is the truth, is contained in the interval. But for the red intervals, they actually do not contain that orange value 0, which means they miss. Those intervals fail to cover the value you're looking to approximate. And there's no way of knowing if you have a good or a bad interval. You just know that in this case, 95% of them should. So in this certain picture, 95% of these lines here actually do cross through 0, but 5% of them won't. And there's no real way of knowing if you're in that what case your interval is in. You can only provide a certain amount of confidence towards that. But what you can do to be more sure is that you can try to increase your sample size, because the margin of error typically will shrink as the sample size increases. And that's because the margin of error is made up of just a constant multiplier, but then the standard error of a statistic. So the standard deviation of a statistic may not be influenced by the sample size, but the standard error of the statistic, which is something different, which is kind of the spread of the mean versus just the spread of the original data, that often will shrink with the increased sample size, which is why as experimenters, you try to get as large of a sample size as possible.
So I'm going to be talking a little bit about a brief overview, rather, about hypothesis testing. So in hypothesis testing, the general flow is to essentially create or devise two hypotheses, your null and alternative. And then based on that, based on whether you assume the null hypothesis is true, you calculate a standardized statistic, calculate a p-value, and then go to a conclusion. So step by step, when you create your hypotheses, your null hypothesis first. It is essentially your unknown population value, which equals some value. So oftentimes it's just set to 0. And your alternative is basically nearly the opposite, the unknown parameter, which does not equal that said value in the where you set your null hypothesis to. So next, when you assume that the null hypothesis is true, assuming that the null hypothesis is true, you calculate a standardized test statistic, and then using that, you then calculate the p-value. And this p-value will help you determine like how likely or the probability of the outcome, like how probable is it that this outcome will occur. And based on the assumption that your null hypothesis is true. And the way you can recognize whether you should be rejecting the null or failing to reject it is figuring out if your p-value is sufficiently small. And normally the p-value, as long as it's less than 0.05, you would reject the null hypothesis. Otherwise, you fail to reject the null hypothesis. And in hypothesis testing, one of the things that can occur are errors, which I will talk about in the next slide. So hypothesis testing outcomes. So there are two main errors that occur during hypothesis testing, or especially when it comes towards the conclusion, is when you do, for example, a type 1 error, which is also considered a false positive. That is when you reject your null hypothesis when in actuality it is true. So to better explain it, on the top right corner, there's a nice little graphic that kind of goes through the types of errors that could happen during hypothesis testing. So on the top right box, you see type 1 errors where you reject the null. That's the conclusion from the statistical analysis, but the true nature of that hypothesis, the null hypothesis, was actually true. And these are also considered false positives, which an example I could give is in medical testing. So I'm pretty sure a lot of you are familiar with when you go get a COVID-19 test, and you either get a positive or a negative result. In this case, false positives, that basically indicates that when a test comes through and it says that you are positive when you really are actually negative. And on the opposite end, type 2 errors is essentially when that test comes back negative, but you are actually positive. And so as I segued into type 2 errors, that in statistical terms is when you reject or you fail to reject the null, but it is false. So again, in the little graphic on the top right corner, on your type 2 error, you can see on the top, it says accept the null, but in more like proper statistical terms, you actually fail to reject the null when the null hypothesis is actually false. So these are two very common errors that can occur in hypothesis testing, and more commonly type 1 errors. So another part of hypothesis testing is sensitivity versus specificity. So sensitivity is regarding the true positive rate. So that is essentially looking at how often that null hypothesis is rejected when the null is false. So how many times are you actually getting the correct outcome based on your hypothesis test? And specificity is, again, the opposite, where it's looking at the true negative rate. So how often we fail to reject the null when the null is actually true. So how many times are you correctly finding out through your hypothesis test that this is indeed negative and you are getting it right? So that is the difference between sensitivity and specificity in hypothesis testing. And so these are the 4 possible types of results that could come out of a hypothesis test that you should always be aware of. So in the next part of our workshop, we're going to be looking more in depth into common techniques in research. And as you can see by our 2 logos, we're going to be looking at SPSS software and RStudio software. So first, I'm going to go a little bit into some of the basic techniques that are used, or basic statistical techniques that are used in research. So the first one, which I'm pretty sure many of you have heard, is linear regressions. So it is essentially a method to model the relationship between a dependent variable and an independent variable. And underneath linear regression, there are 2 types. There is simple linear regression, where you are finding a relationship between one dependent variable and only one independent variable. Whereas in a multiple linear regression, you're looking at the relationship between one dependent and more than one independent variable. So for example, a simple linear regression will be an example that I put there is gas mileage, which would be an independent variable, versus driving speed, which is a dependent variable. So you're looking at the relationship between how gas mileage affects someone's driving speed. In multiple linear regression, my example that I've seen throughout, and it's pretty common throughout many research, or at least I've seen in common weather reports, is rainfall, which is our dependent variable, versus the two variables that is affecting it, elevation and temperature. So those would be our two independent variables in this case. And also, I do understand that there is definitely more than two independent variables for rainfall, but these are just the two I listed as an example. And one of the main things that we see a lot in research in terms of linear regressions is transformations. And this involves maintaining that linear relationship between your dependent and one or more independent variables by changing the variables only by a constant value. And this basically maintains that when you do your line of best fit that I'll show on a next page on an example graph, that your values maintain as close as possible to your best fit. In a nonlinear transformation, however, this involves the linear relationship being changed by manipulating the variables through square root or reciprocals. So the difference essentially is that one is maintaining the linear relationship, the other one is not. In logarithmic transformations, which is another common type of linear regression transformation, is taking the log of x while maintaining the linearity. So this one's a little interesting because while it is changing the variable somewhat similar to a nonlinear, it still actually maintains the linearity of that relationship. So there are definitely many more examples, and I definitely suggest taking a look at those as well. But these are just a couple that that have come out commonly in many types of research. So as I said before, I was going to show an example. So this is from an odontology paper, which interestingly is basically a scientific study of teeth and diseases regarding teeth. So this, for a little bit of background, this paper was looking at how fluorescence intensity basically affects color coordinates on imaging. And they were trying to figure out how much fluorescence intensity would they need to get better images. And in this particular graph, for example, so your fluorescence intensity would be your independent and your L asterisk values would be your dependent variable. So this kind of shows an example of how a line of best fit is put onto a regression graph, and you can kind of see how close the values are to that line of best fit.
Okay, so in most situations in research, you'd look to use some type of regression model because it's very easily interpreted. And it's a good thing in research to be able to interpret the results you have. But linear regression is best used when your data that you collect is on a continuous scale. But sometimes you do research where your variables that you're looking at aren't really on a continuous scale, but are rather discrete categories. And the appropriate situation that's often favored in research when you have categorical variables and a quantitative response is what's called the analysis of variance test, or ANOVA. And what it does is it tests for significant differences in the different treatments of your independent variables. So for instance, just imagine you had a flyer of 3 different colors. It was red, blue, or green. And you wanted to see if the number of people who filled it out, the response rate changed based on the color that it was. So you could do an analysis of variance to determine if there is any relationship between the response rate and the color of the flyer. In that case, color is not something that is on a continuous scale. It's just discrete categories, so it's an appropriate thing to use ANOVA for. Linear regression could be used in this setting, but it's not really favored, and ANOVA is just the preferred method. And typically, you'll see one-way versus two-way ANOVAs. Anything more than that becomes a bit too complex, but a one-way ANOVA is like the example with the flyers. You only have one variable you're looking at, which in this case would be color. But you can also have two-way ANOVA, where say you have color and maybe if it was laminated or not. So you could have 2 different variables that you're examining together, and you could test for the way those 2 different variables interact. And ultimately in ANOVA, your default assumption is that there is no relationship between the treatment levels that you're looking at and your outcome. But if you do find a relationship based on your hypothesis test, you can do follow-up analysis. And the most commonly used follow-up analysis is known as Tukey's method to determine the pairwise differences between treatments. So for instance, if you're doing your flyer example and you found that there was a significant relationship between the color and the response rate, you could use Tukey's method to determine if red had a higher response rate than blue, which is higher than green. So once you determine that there is some type of relationship there, you can use follow-up analyses like Tukey's method to determine the extent of those relationships. So here is a Anova table from an actual article in a journal. This is Journal of International Business and Economics. It was looking at a type of development index as the outcome variable. And their independent variable, their category that we're looking at, was a type of score that they've generated called like a security index. So they're comparing the low and high security indices as their treatment levels for the factor of the security index. And the purpose of ANOVA is to kind of partition the variability into the different categories to get a p-value, in which case this p-value is quite large, so they would fail to reject the null hypothesis, going back to the hypothesis testing. This is another figure derived from Anova in a article. So this one is a drug study, in which case they have two different types of genes shown by the red and the blue bar graphs, and a certain dosage of a drug as a different factor levels. So they have two different drugs, each of 4 levels each, for a total of 8 different combinations. And this is typically how you might see ANOVA in a scientific paper. They determine the bars, the link between the bars and the asterisk determines the extent of their significant differences, which is determined using the two-way ANOVA and the follow-up Tukey's method for analyses. So I'll be talking about R and giving a quick demonstration of it, but what R essentially is, it's a statistical programming language using vectors, kind of like MATLAB. So a vector is essentially just an n×1 matrix. It's just a list of values. And it's very convenient because any operation you do will be done to every element in that vector. It's very convenient to use for simulation studies, but at the same time has all the type of functionality you might want in your other programming languages like conditional statements or loops or function definitions. And another usage is its ability to make plots. So now I'll just be showing a quick demonstration of how to load in data and run an Anova and regression example. So here's my RStudio. RStudio is the way 99% of people access the R programming language, and it's free to download. When you first open it up, you see kind of 4 quadrants. The bottom right is your file directory, where you also see your plots. The top right is where you'll see any of the variables you define. The bottom left is your console, where you can type in any command you want, like 2 + 2 here, and it'll give you an output. And the top left is where your actual code editor. So in this case, I'm going to show you an example of loading in data. So if you have a CSV file in your file section right here, in this case, I have cook.csv. I can use this read.csv command to load in the data, and R will generate that for me. And if I look at it, I very easily have this data set in R that I can manipulate much faster than I would be able to in Excel or another software of that nature. So I can use the str function to determine the kind of the nature of that data set. So it'll tell me I have a pot, which is a factor, or a dish, which is a factor, and then I can use the head function to print out the first couple of data sets to make sure it all looks what I'm looking for. Just to give some background on this was from a study using a two-way ANOVA, in which people were looking at the iron produced in food in different kind of conditions. So one of the conditions, one of the variables was a type of pot that was used, whether it was aluminum clay or an iron pot. And the other factor was the type of dish, whether it was a meat, legume, or vegetable dish. And they're measuring the amount of iron in micrograms in the food. And just for some quick demonstration, once you're doing this, you have 6 different possible combinations of these levels, and you might want to know the mean response in each of those levels. And within R, it's only a matter of a couple lines. This function itself makes all the means, and then I just plot it into a matrix here. And I can see immediately all the means without much hassle which is very useful for my ANOVA analysis. And there are also some built-in plots which are used with the ANOVA. I can use this plot.design command to create a plot, which shows me the different means of each of the 6 levels and then the overall mean, and even an interaction plot to test if there's interacting behavior between the type of dish and the type of pot. But more importantly, you can use this AOV function to generate the Anova table, just what you saw on the paper there. And if I use the Anova command, it'll generate this table where it will show me my p-values, which is what I use to determine whether or not there's a relationship between the variables. And then just for, you can do a linear regression with this LM function. You do the y variable and then a tilde in the x variable. So in this case, I have miles per gallon against horsepower of some cars. Here's a summary of my R-squared value, and I have the values of the coefficients. And you can very easily plot the line of best fit. So here I use the plot command to plot the x and the y values, and I set their labels, and I add the line of best fit very simply. And just to very quickly, last thing to go over the box plot and histograms we talk about, there's a default box plot command in R. You just give it a list of data, and it'll create a box plot for you. Or you can use a histogram just with the hist command, which you can, customize with the color, the name, anything you like. I can make this blue in UF Spirit, and you can see the blue bars there. And, with R, you can get more sophisticated types of plotting. You can generate multiple plots on the same visualization. And all of it is very customizable, and it's very easy to use once you get the hang of it. So I'm a big fan of the software.
Thank you so much, Brett. That was a really good explanation and demo of R. So the next software that we're going to look at and Mark will demonstrate will be SPSS.
Okay, so SPSS is just another commonly used software package that is used for batch statistical analysis. What this means, batch statistical analysis, is that you can directly input a workbook, like an Excel sheet or a .csv file, directly into the software, and it allows for an auto-population of all your cells and rows to go directly into the software. As you can see from the screenshot on the right there, you have all of your row names, and you have, as well as all of your column names with all of the specific data that you have in that CSV or Excel sheet. SPSS is extremely convenient just because it requires little to no prior knowledge, and it's very user-friendly because you actually don't have to do any coding. It's really just selecting data and clicking, as we'll show in a second. And then lastly, it's actually available to all UF students through UF apps, and that's actually where I'm going to show it from. Okay. So on the screen, you can see that this is actually some of my personal data from the research that I do. And just to give a little bit of some background context on this, this is just 12 different Parkinson's disease patients. And I am analyzing their gait or their walking during their first 50 strides, and I'm comparing their data to the last 50 strides. I have all of their data here for a multitude of different types of variables, but I have mainly the most important thing is that I have the mean and the standard deviation for all the different variables here. And one of the things about SPSS is that you do have to arrange everything row-wise or column-wise, excuse me. And what this means is that all of your subjects actually have to have all of their data on the same row. And then all of the columns have to correspond to the same set of data for this for the different participants. And what that means is that you just basically have to line up, in this case, sorry about that. In this case, you are actually looking at the first 50 strides for these people, and you just want to make sure that each one of these data sets corresponds to that specific subject for the specific variable that you're looking at. What we're going to do today is just a simple t-test or a repeated measures or a paired samples. So with SPSS, after you import your data, and the way that you do that is you click File, Import Data, and you can go ahead and go to any of these data types that you're trying to import and open them directly from your computer into this data sheet. After you have all of that populated into the software, you want to go to the Analyze tab. And because today we're just going to be looking at a paired samples t-test, also known as a repeated measures t-test, we're going to look at either compare means for a paired samples t-test, or you can go to general linear model for repeated measures. One of the good things about SPSS is it can do all of the different types of statistical analysis that you need. There's also correlation here, there's regression, and you can also make graphs and all different types of figures on here. But just for time's sake today, we're just going to look at one specific thing. And here. So after I click paired samples t-test, what I'm going to actually look at is I'm going to be comparing the propulsive force during the first 50 strides of the Parkinson's disease patients to the propulsive force of the last 50 strides for the Parkinson's disease patients. And as you can see here, I just paired up the first and last variable, and all that is designating is it's going to be doing a statistical analysis or a paired samples t-test between the first 50 strides for all of these participants right here, as well as the last 50 strides, which I have right here. And it's actually comparing the means and seeing if there's a significant difference between them. So after we have this one pair set up, you can continue to go through all of the other data here and continue to pair them to their appropriate pairs. But just for time today, we're just going to look at one. So after you pair those two together, all you really do is click OK. And one of my favorite things about SPSS is that it spits everything out into this PDF-like file that is extremely easy to read. And it shows you exactly what you are comparing in your statistics. And it also, because for this test, we're looking at a paired samples test, you can go down to the bottom and you can scroll over. It gives you your significance, also known as your p-value, your degrees of freedom, and your t-value. In this case, we had a significance or a p-value of 0.109, which was found to not be significant. And once again, you can directly save this file and you can have it open at any time to go back and look again. And yeah, so that is mainly all that I had to talk about for SPSS today. And I believe Kavitha has a few things to say about the end of our workshop today.
Yes, so that will be the end of our workshop. But before you go, please, before you go, we would like for you guys to fill out a feedback survey that either Mark or Brett can share in the chat real quick. Essentially, it's just going to ask about, you know, what did you think about it? What would you like to see next time? Things like that. Also, make sure to follow us on Curbs or follow Curbs on social media. We have an Instagram at UF-Curbs and we have a Facebook at UF-Cur. If you would like to come to more workshops like this, you may sign up for the listserv. Our email is curbs, all caps, at aa.ufl.edu. And other than that, yeah, Ali just put the end of workshop survey in the chat. So if you guys wouldn't mind to please fill that out and give any feedback that you have for this workshop. With that, does anybody have any questions you would like to ask us at all? You can just unmute yourself or put it in the chat too, and we'll make sure to answer them.